The human genome plays an important role when it comes to studying the history of humankind. Indeed, it's encoded in the DNA of every human being. It contains information not just about a person's appearance and diet, but also about their ancestors, even thousands of years after their death. Comparing the genomes of various living beings provides information on their degree of kinship. Family trees are used to illustrate these kinships, and they can also be drawn up for related species. The molecular clock is helpful in dating the branches of these family trees. As a timepiece, it uses the mutations in DNA that accumulate from one generation to the next. What is DNA? Our entire genome is contained in virtually every cell of our body. Our DNA is spread over 23 pairs of chromosomes. It is a thread-like, tightly wound molecule. It contains the instructions for the complete structure of an organism with all its biological functions. DNA consists of varying sequences of 4 genetic letters. Nucleotides with the bases cytosine, C, guanine, G, adenine, A, and thymine, T. A comparison of the genome of any two persons shows that their DNA is approximately 99.9% identical. The tiny differences are due to changes in the DNA passed on through generations. These changes are called mutations. However, the way the differences between individuals are distributed is also determined by new genome compositions. Such recombinations occur during reproduction. Mutations and recombinations are the drivers of evolution. Deciphering them allows us to specify more accurately the chronological evolution of human beings and other species. How can mutations and recombinations in DNA be used as a clock? DNA mutations occur in all living beings. They happen during cell division as a result of external influences or quite spontaneously without any apparent reason. Most of these mutations are instantly repaired by the organism, but some are not. Fortunately, the vast majority of mutations are neutral, that is, they are not even noticeable. But others can cause illness. Very rarely do mutations have a positive effect. A mutation can be passed on to the next generation, with further mutations then occurring in its DNA. In other words, these mutations increasingly accumulate. The longer it is since living beings had a common ancestor, the greater the differences. So if we wish to study stages in evolutionary history that go back a long way, mutations are an obvious choice as the starting point for the molecular clock. The differences that have accumulated over a long period of time are used, for instance, to estimate the point at which Neanderthals and modern humans began to evolve along separate branches. Their last common ancestors date back to more than 500,000 years ago. The molecular clock was similarly useful in determining the demarcation between chimpanzees and modern humans. They began evolving along different branches of the evolutionary tree some 6 to 7 million years ago. DNA mutates at different rates in different species. However, each species has its own distinct mutation frequency, referred to as the mutation rate. The mutation rate can be derived by comparing the DNA samples of different ages from one and the same species. Radiocarbon dating, for example, is a method well suited for reliably dating DNA. It can be used to determine how old a bone is. DNA is taken from the bone and compared with the genetic material of, say, a modern human. The mutation rate can then be estimated based on the missing differences in the old genetic material compared with the new. More recent events in evolutionary history are better estimated using recombinations in the DNA. In humans, recombination occurs during reproduction when pairs of chromosomes from two individuals combine. Each inherited chromosome now contains a combination of segments from the two parental chromosomes. This process occurs in every generation. It means that, with each generation, the segments that match in two persons with a common ancestor become shorter. The number of recombinations in one and the same species remains largely constant for each generation. This number can then be used to calculate when the last common ancestor of two individuals lived. The method has been used to show that there were also Neanderthals among the human ancestors who migrated out of Africa many thousands of years ago, and that the species of humans living today interbred with Neanderthals some 50,000 years ago.